Welcome to UMDF's Ask the Mito Doc monthly webcast. Tonight we will hear from Dr. Mary K. Koenig from UT McGovern Medical School in Houston, Texas, and Nicole Wilson, UMDF's Program Manager with the Science and Alliance Department. Our moderator for the Q&A this evening is our very own Margaret Moore, UMDF Support and Education Associate. Let's get started, Nicole.
Hello, everybody, and welcome. We just want to take this time to thank our sponsors for today's webinar, Edith L. Trees Charitable Trust and UCB. We thank you so much for all of your commitment and help and your ongoing support. All right, so we're going to go ahead and get started. And as Kara did a little introduction, I am Nicole Wilson, UMDF's Program Manager for the Science and Alliance Department. Today, I just wanted to talk to you a little bit about some of the programs that we have going on. And next Wednesday, you can join Philip Bieski and myself as we share our first year findings about MitoShare. So we're going to talk to you about what's important about joining a patient registry and how that's important aiding in your diagnosis as well, and also helps you learn more about some clinical trials as well. But some really great things in there. And this is going to be a really fun one too, because we're going to show you what we learned. So some of those things of what we learned is that we realized that 25% of the people who are in MitoShare are not diagnosed. So those are people who are still going through their diagnostic journey. And our next program is really important to help those 25% that haven't. And that is our genetic testing program that's financially supported by Reneo Pharmaceuticals. And this is a no-cost genetic test program. This test can only be ordered by your clinician. So it's really important for you to share this information with them so that they can get a test kit ordered for you if you haven't had genetic testing done yet. The only eligibility that they have is that one, it's your clinician that's ordering it for you and that you both live in the United States. You also must have a type of clinical diagnosis where the doctor feels it's consistent with a primary mitochondrial disease and that you've never had any type of genetic testing done before. And you've exhausted all of your options with paying for that via insurance, which we know a lot of our patients do sometimes have struggles paying, getting their insurance to cover that genetic testing. So this is a great program for you. So please make sure you pass that along to your physician to order for you if you haven't had genetic testing done yet. One of the next programs that we have is our grant program, and we just wrapped this up on February 27th. These are all now in peer review by our Scientific and Medical Advisory Board. And some of the grants that we are giving is for our postdoctoral fellow, which is our accelerator program. If you're not familiar with that, that's a household that donates a total of $500 or more throughout the year, then you're eligible to give a vote for that accelerator. So it's a really exciting way that you can really help fund research. Another one is our principal investigator, which is $100,000. And a new one this year is clinical trial readiness. And that was out of a $50,000 pool. So really exciting. And all of those awards are going to be announced at our symposium. That's coming up on June 28th through July 1st. So we are starting sessions on Thursday. So hopefully you're able to join us early instead of just our normal Friday, Saturday. We are doing some Thursday afternoon sessions, and we will be in Charlotte, North Carolina. Scholarships are available. That deadline is April 15th. So if you haven't already submitted an application for a scholarship, please go ahead and do that. You can do so at umdf.org/symposium to find the form. And we also have some really great volunteer awards. So if you know somebody who's really committed themselves to their energy to mitochondrial disease and UMDF through either fundraising or advocacy, or even just letting other people know how great UMDF is and really talking to them about mitochondrial disease, then you can nominate them for this amazing volunteer award. It's also another great place to meet a lot of other mitochondrial disease families and get to chat with some of our clinicians. One of those clinicians is our speaker today, and that is Dr. Mary K. Koenig. Dr. Koenig, welcome. Thank you so much for joining us this evening.
Thanks, Nicole. I'm really happy to be here, and I'm honored to be talking to everybody tonight. Let's see. Okay, so again, I just want to say thank you to the UMDF for giving me this opportunity to talk with everybody. I am going to be talking with you guys tonight about emerging therapies for primary mitochondrial disease. I think this is a really exciting topic. It's interesting, this topic came up for me last year, kind of towards the end of the year in the fall. I was asked to go to Australia to talk at the AHSCT conference, and this was the topic that we settled on. And I got really excited looking at how much has been happening in the world of treatment and therapy and advancements for mitochondrial disease recently. And so I've kind of repurposed this talk a couple of times since then, and I was really excited to have an opportunity to redevelop it, to talk with some of the patients and family members about it. So I'm going to talk with you guys today or this evening about some emerging therapies for primary mitochondrial disease. So one of the things that I think is extremely important for us to remember is that mitochondrial disease is a relatively new concept. As much as mitochondria influence our lives, the concept that they could be involved in disease is actually a relatively new one. The first patient that was believed to have a medical condition secondary to dysfunction of the mitochondria, or the first person that was identified as having a medical condition secondary to a problem with the mitochondria, was a young woman with what's called hypermetabolism. And this was described in 1962 by Rolf Luft and his colleagues in Stockholm, Sweden. And that really wasn't that long ago. More disorders were identified throughout the 1970s, and people started understanding and localizing more that there was defective mitochondrial function and that it could be associated with medical disease. In the early 1990s, we continued to understand that there was a multisystemic nature to mitochondrial disease, and also that there was this genetic control, and that it was coming not just from the mitochondria themselves, but that the nuclear genes, the regular or typical genes, could also be involved in mitochondrial function and mitochondrial disease. And then for the rest of the 90s and through current day, we've continued to understand mitochondrial disease genetics, and we've started identifying multiple new genes that are associated with mitochondrial disease. And we've learned a lot about how the mitochondria function and what happens when they dysfunction. So kind of my point with this is, for the last 60 years or so, we've just now started recognizing, one, that mitochondria can cause disease, and two, how they do it. And we've started trying to describe what kind of diseases there are, how they affect people, and really understanding how those diseases are inherited, how they are affecting the mitochondria within the cells, and what's actually going on in patients when they have mitochondrial disease. Okay, so the first thing we're going to look at is the electron transport chain, oxidative stress, and mitochondrial biogenesis. Okay, and what I've tried to do is kind of group us into different targets that we're looking at for therapy with mitochondrial disease. So one of those targets is the electron transport chain and ATP function. So when you're inside your mitochondria, the electron transport chain is what we use, or what we think of, that takes kind of our basic building blocks that we extract from our food, things like what we get from our sugars and our fats and our proteins, and converts it into the chemical energy in the form of ATP. And whatever the cause of a person's mitochondrial disease, one of the commonalities that we see in patients is that there is a problem with the function of the electron transport chain. So although all people with mitochondrial disease seem to have their own unique mitochondrial disease, and they all seem to have very different symptoms, one of the commonalities they do have is this problem with electron transport chain function, and ultimately a lack of production of enough ATP. And so this lack of ATP production, or lack of enough ATP production, is what generates a lot of the symptoms that we see in patients with mitochondrial disease. So early on, it was thought, hey, if we can improve the efficiency of ATP production, maybe we can make the symptoms of someone's mitochondrial disease better. And it was actually one of the very first targets that was considered as a treatment modality in mitochondrial patients. And I promise I'm going to give some examples. So this is a picture, a very complicated and yet very simple picture of what the electron transport chain looks like in a textbook. It's actually a very complex chemical process, and it requires hundreds and hundreds of moving parts in order to work correctly. And the reason we say there are so many different types of mitochondrial disease is because a dysfunction or a problem with any one of these parts can cause the mitochondrial disease. But ultimately, again, over here, it's the problem with ATP production that's the commonality. So one treatment concept that people have come up with is, let's give people more parts. If they're missing some parts or the parts aren't working well, maybe if we supplement some of these parts, we can make the electron transport chain work better, work more efficiently, work faster, and maybe we can make it produce more ATP. So another concept that we think about in mitochondrial disease is something called oxidative stress. So free radicals are molecules that get created within the mitochondria and within the cells as byproducts of normal things that are happening inside the cells. So everybody, whether they have a mitochondrial disease or not, creates free radicals in their cells. Poorly functioning mitochondria overproduce free radicals, and free radicals damage other things inside the cells. And this is what we mean when we say oxidative stress. So in everybody, we have some degree of oxidative stress, and there are some things that increase oxidative stress. Things like smoking and drinking heavy amounts of alcohol can increase your oxidative stress. They cause more production of free radicals. This damages your cells further, and it can cause damage to some of the things within your cells. And so a person with a mitochondrial disorder will overproduce free radicals within their cells and can damage their cells from this oxidative stress. So one of the treatment theories that was developed is, well, let's give people antioxidants, try to protect against some of this oxidative stress, try to reverse some of it, tamp down or dampen down some of these free radicals, and see if we can relieve some of the oxidative stress to improve the health of their cells and their mitochondria. Another concept is mitochondrial biogenesis. So let's just make more. If we have a mitochondria that produces half the amount of ATP that it's supposed to, let's make 2 of them, and then we'll have 100% or a normal amount of ATP produced. And so mitochondrial biogenesis is a concept wherein we say, if we can't fix the mitochondria and make them work normally, then maybe we just make more of them, and maybe more of them produces enough ATP to make the cells function properly. Now, the problem with that is that, again, everything within our bodies is very complicated, and there's a whole lot of signaling processes that occur within your cells that are controlled by genes, both in the mitochondria and in the nuclear DNA. And it's not as easy as just making an extra mitochondria. But there are ways to do it, and it is one of the targets that people are studying now. And there have been some studies done in animal models that show that if you can increase the number of mitochondria, you can sometimes improve the function of the animal models with mitochondrial dysfunction. So let's look at some easy examples or some simple examples. So when we talk about supplementing factors in the electron transport chain, one of the easiest things to look at is NADH. So NADH or NAD plus plays a role in the electron transport chain. It's one of those chemicals that functions, or one of those products that functions within the electron transport chain. And so some people take supplemental NADH. And so higher levels of NAD also are known to activate mitochondrial biogenesis, and they also serve as antioxidants. And so a lot of doctors and a lot of patients will take supplemental NAD or NADH to try and increase their electron transport chain function, activate mitochondrial biogenesis, and also help as an antioxidant. Niacin is another molecule that a lot of people take, or another vitamin that people take. It's a precursor for NAD. It's going to work in the same manner. And there have been some studies that have shown that there may be some benefit to taking those. The problem with a lot of the supplements is that they haven't been studied very rigorously. And so there's not a lot of data one way or the other to show which ones work for which patients. CoEnzyme Q10 is another one. It's a naturally occurring molecule. It's found almost everywhere. It's part of the electron transport chain. It's also a very significant antioxidant. And it can lead to a lack of it can lead to poor ETC function, can lead to oxidative stress. And again, although we widely recommend it because there's a lot of theoretical reasons why it should help, there hasn't really been any conclusive studies showing that it helps. Taking it one step further, the molecule adenine is a synthetic analog of CoQ10, and it has been studied in labors and in meat loss, and there have been some studies showing some clinical benefit in patients with LHON or labors who have received this. So here's a list of just some other compounds that are under investigation as antioxidants or for mitochondrial biogenesis. And this is by no means meant to be an inclusive list of everything that's being investigated, but these are some things that people are doing studies on, and you can easily find these by going to one of the clinical trials websites. And I know that we're going to put up some of those links for you guys later where you can look at those. And this is their mechanism of action, either antioxidants, mitochondrial biogenesis, or a combination. Okay, so let's switch away from those and let's talk about another potential target. So what else could we look at to help patients with mitochondrial disease function better? So one of the other things we look at is nitric oxide metabolism and vascular integrity. There are certain types of mitochondrial disease that are associated with dysfunction or lack of function of the blood vessels. The one that we think of in particular is meat loss, where patients will develop stroke-like syndromes. But there's a possibility, and there's a lot of talk about whether or not some of the cardiovascular complications of mitochondrial diseases can also be related to vascular integrity, and whether or not there's some of the similar abnormalities seen in patients with cardiovascular type diseases as well, or whether some other complications of vascular nature could be associated with a similar mechanism. So this starts to get really complicated, but the simple version of this is that people need nitric oxide in order to make their blood vessels stay open. And if your blood vessels close off, then the blood can't flow through them. And if the blood can't flow through your blood vessels, then you end up with a stroke or a heart attack because the blood doesn't get where it needs to go, and your brain or your heart or wherever else in your body doesn't get what it needs. And so we need a certain amount of nitric oxide in our body in order for your blood vessels to stay open so the blood can flow through them. And in times of metabolic stress, some patients with mitochondrial disease will try to increase the production of their mitochondria, and they will upregulate the production of complex 4, which is part of the electron transport chain, or cytochrome C oxide. So we were talking about nitric oxide. Hopefully we got the basic of nitric oxide. And so we need nitric oxide because we need it to keep our blood vessels open so we don't have strokes or heart attacks. And we know that L-arginine is a precursor for nitric oxide, which means that it helps us make nitric oxide. And there have been a lot of studies that have shown that L-arginine can prevent strokes in patients with meat loss, and that reversing a depletion of nitric oxide can improve blood flow. And then there's the question of citrulline. Well, citrulline is also part of this pathway that forms nitric oxide and L-arginine. So citrulline and L-arginine help us form nitric oxide, which helps us increase the blood flow in patients with mitochondrial disease who may be depleting their nitric oxide because of their mitochondrial disease. Okay, so what else do we think about? We think about lactic acidosis. So lactate overproduction is well documented in some types of mitochondrial disease. We have some patients that develop lactic acidosis, and increased levels of lactic acid can produce a lot of different deleterious or dangerous effects on the body. And so one of the things that's been studied is how do we get rid of the lactic acid, get it out of the body, to try to make people feel better. So increased lactic acid in your body can cause muscle cramping. It can cause trouble breathing. It can be dangerous if it gets high enough because it can make your blood actually become acidic. And so there have been several medications, things like dichloroacetate, phenylbutyrate, that are under investigation as options to try and reduce lactic acid levels. And then a ketogenic diet can be used in specific situations and can also reduce lactic acid levels. Structural integrity of the mitochondria. So one of the things we know, and I'm going to look down here at these pictures, one of the things that we know about the mitochondria is that they have to maintain their structural integrity. It's kind of like everything else. If you have a cup, for example, that you're trying to keep water in, and all of a sudden the cup starts changing the shape of it, the water is not going to stay in there very well. And you need to be able to maintain the structure of that cup if you want to hold water. So the electron transport chain sits in the membrane of the mitochondria, and the structure of that mitochondrial membrane is very important to how that electron transport chain works. And it's been shown over and over again that in people with mitochondrial disease, they lose that structure in their mitochondrial membrane. And so there are drugs that are being developed that are targeting that membrane structure and trying to normalize it and trying to bring it back to a more normal mitochondrial structure. And these drugs have been shown to help increase ATP production. So DNA modifications. I hear every day about gene therapy, and people always ask me about gene therapy. And so there's a lot of different types of gene therapy, that can occur. It's not just a single thing. So one of the things that's been really talked about lately in people with mitochondrial DNA disorders is something called mitochondrial augmentation therapy. And mitochondrial augmentation therapy is very interesting. So it goes by the concept that most people with mitochondrial disease have a mixture of cells inside their body, or a mixture of mitochondria. Some of their mitochondria are healthy and some of them are not. And depending on what that ratio is, it can influence how severe their disease is. If they have more healthy mitochondria, they have less severe disease. And if they have more unhealthy mitochondria, they have more severe disease. And so one of the concepts has always been to try and shift that back to more healthy mitochondria and less of the unhealthy mitochondria. And mitochondrial augmentation therapy is really driven to try and address this concept of, can we shift away from the unhealthy mitochondria? So what's being done with mitochondrial augmentation therapy is patients' stem cells are being removed from their body, or hematopoietic stem cells are being removed, and they're being grown up, and they're being enriched with healthy mitochondria. So basically what you're doing is you're taking a patient's own stem cells that have this mixture of healthy and unhealthy mitochondria in them, and you're adding to them healthy mitochondria. And you're growing them up, and you're changing this ratio of unhealthy to healthy to favor healthy, so that there's more healthy mitochondria within the patient's own stem cells, and then you reinfuse them back into the patient. And this is called an autologous process because you're using the patient's own stem cells. And so it's pretty exciting because the process is, again, it's autologous. And so there's very little risk for things like a transplant rejection. And so you're taking a patient's own cells and enriching them with healthy mitochondria and then reinfusing them back into the patient to allow them to disseminate throughout the body. And there are currently studies underway looking at this therapy and how it will work. But right now it is under investigation for mitochondrial DNA disorders, those that are inherited from the mitochondrial DNA. This is another one that I found to be reasonably interesting, also for mitochondrial DNA disorders, something called Mesangioblast Therapy. So mesangioblasts are a special type of stem cells that come from the muscles, and they often contain a lower heteroplasmy level than cells found in other tissues. And so what's being done with this therapy is that mesangioblasts are being collected from a patient who has a mitochondrial DNA disorder, and those with lower heteroplasmy levels are being selected. So just taking the patient's own stem cells, and then they're growing those stem cells up in the lab, the ones that have lower heteroplasmy levels, and re-injecting them back into the patient. Again, with the theory of providing them with stem cells that have higher levels of healthy mitochondria and lower levels of unhealthy mitochondria. Also still under investigation. Nuclear gene therapy. This is more the classic gene therapy that people think of. And this is where you take a gene and you replace the dysfunctional part of it. So what they're doing is, and this is being looked at in LHON right now, is they're taking a virus, a very small virus, and they're putting a healthy gene in to the virus that is the same as the broken gene in the patient with LHON. So the patient with LHON has a genetic change that causes them to have LHON. And so what the investigators are doing is they take a virus and they find a healthy gene that has the normal function of that LHON protein. They put it in the virus, and then they're injecting it directly into the eyes. And then that new virus goes into the eye and it produces the protein that it's supposed to produce, because the patient can't produce it. Their gene doesn't work. And so they can't produce what they're supposed to. And so the virus produces it and it competes with the bad protein and ends up restoring the function for the defective gene. So it's not a replacement. They're not replacing the gene. They're just adding a healthy one and allowing it to make functional protein that's competing with the dysfunctional protein to restore the function within the eyes. Okay, so let's look at nucleoside replacement therapy. This one has also been a pretty hot topic lately. So mitochondrial DNA maintenance. So everybody has regular DNA inside their bodies. And then we also have a smaller piece of mitochondrial DNA. And some mitochondrial diseases result because the mitochondria can't properly copy their mitochondrial disease. Their mitochondrial DNA. If it makes you feel better, there are also other genetic diseases in people who can't copy their nuclear DNA, but they're not mitochondrial diseases. So there is a subset of mitochondrial diseases where patients are not able to copy their mitochondrial DNA appropriately because they can't form the proper building blocks called nucleotides that are needed for mitochondrial DNA synthesis. And so there has been a treatment developed with exogenous nucleotides. So these are nucleotides that are synthesized outside the body and administered to the patients orally as a medicine. And it has led to improvements in some specific conditions, and it's under investigation for other disorders of mitochondrial DNA maintenance. So this is kind of a special group of disorders, and there's a special group of treatments that are being investigated for this group of disorders where they're treating them with these nucleotides. Enzyme replacement therapy. So enzymes are proteins that are in our bodies and they help chemical reactions occur. So it's kind of like adding, a booster to something to make it happen faster. That's what the enzymes are. And if you don't have enough of an enzyme, and it can either prevent the formation of something that you need. So if you need a particular molecule for your electron transport chain to function properly and you don't have the enzyme, you may not be able to do that. Or it can prevent you from breaking down something that damages the mitochondria. So for example, a free radical or something along those lines. So you end up with this accumulation of toxic molecules or lactic acid or something that ends up in your body that causes damage. So enzyme replacement therapy is a treatment that's used to replace enzymes in patients who have a genetic disorder where an enzyme is lacking. And interestingly, there are a lot of metabolic disorders that have been following this theory and using this for a long time, things like phenylketonuria, and some other genetic disorders that have been investigating these types of enzyme replacement therapies for a long time. It seems like the easiest thing to do would be, hey, I'm missing an enzyme, just give it to me. Let me just take it. But there's so much to it. It has to get past all of your stomach juice and get absorbed into the body, and then it's got to go to the right place and it's got to work. And so it's actually a really complicated process to get that enzyme into your body and into the right place so that it'll work. So one of the things that people have done is what's called an allogenic stem cell transplantation. So if you don't have a particular enzyme because your cells are not able to make it, then one of the things that's been done is a stem cell transplant. Replace your cells with someone else's cells that has the functioning enzyme. And this was one of the first things that was tried for people with this type of disorders. And it is effective to permanently replace the defective enzyme in most cases, and it's been shown to do that. However, you're having to replace your stem cells with another person's stem cells who is not related to you. And so it does require full-on immunosuppression, and there is a high risk of rejection and complications. And the mortality rate can be as high as 50% or more in patients undergoing this procedure. So although it is effective and it does work, the mortality rate is really high. And so it's not really considered a very viable therapy. So there's a new process that's being investigated now called Erythrocyte Entrapment, which is really interesting. So what they're doing is they're making the enzyme, and they can't give it to you by mouth because your body would just degrade it and it would never go anywhere. So what they're doing is they're making the enzyme, and then they draw your own blood from you, and they put it in this contraption that causes your red blood cells to open up a little bit. And then they push the enzyme into your red blood cells and close them back up again. And then they reinfuse your red blood cells back into you. And your red blood cells will stay in your body 30, 45 days before they turn over and make new red blood cells. And so what happens is the enzyme stays inside your red blood cells. So say, for example, you need to break down some substance that's floating around in your blood. Well, now you have the enzyme in your blood, as long as you get a treatment every 30 or 40 days to continue to infuse that enzyme in you. And so there's studies looking at that to see how well that can work. And it's called erythrocyte entrapment. Take the enzyme, put it in the patient's own blood cells, and put it back in them. There's no risk for rejection. It's the patient's own cells. But it does have to be repeated relatively frequently because the blood cells only last for 30 or 40 days. Liver transplant. Liver transplant is used in some select situations. So there are some enzyme deficient diseases where the liver is the main source of that enzyme. And so in those situations, there are cases where patients have received liver transplants, and it's similar to a stem cell transplant where you're taking someone else's liver that has a healthy enzyme and putting it in you, and that provides you with a source of healthy enzyme. And liver transplants tend to have much less complications and much higher success rates than stem cell transplants for this type of disorder. So in summary, with all of my complications in my talk, I'm sorry about that. Mitochondrial diseases are still relatively new diseases, but the treatments are starting to advance really fast. I'm super excited about the number of clinical trials that are going on and the amount of research that's being done. And there are a lot of different targets that have been identified and that are currently under investigation as potential therapies. So we're looking at modifying the electron transport chain in the redox state. We're looking at enhancing mitochondrial biogenesis, improving lactate homeostasis. We're looking at the nitric oxide balance, improving membrane integrity, modifying or manipulating the DNA, either the mitochondrial or the nuclear, the nucleoside replacement therapies, and enzyme replacement therapies. The future is truly wide open, and the clinical trials are advancing rapidly. You know, I encourage my patients to look frequently at the clinical trials websites. We try to address clinical trials at most of their visits when they come in and whether or not they're eligible for something. There are more than 50 active interventional studies on clinicaltrials.gov just for mitochondrial patients, and it's probably closer to 70 or 75. You don't need to find all of them. You just need to find the one that's right for you. So I would encourage you to go here or go to the UMDF's website, which is a lot easier to navigate than clinicaltrials.gov, and just look and see if there's something there and if one of these might work for you, and if you're interested in participating. And I want to thank all of you for your participation. I want to thank the UMDF for giving me this forum to talk to you and allow me to disseminate information to more than just the people that I see in my clinic. I want to thank the Mitochondrial Medicine Society for continuing to work to educate physicians. And obviously I want to thank McGovern Medical School and the Center for the Treatment of Pediatric Neurodegenerative Disease. So thank you all, and I'm happy to answer any questions.
Okay, great. I think there's definitely some questions that we have for you. So Margaret, if you want to take it away and get to...
I am ready. Of course, my neighbor's dogs decided to bark at this very moment, but I will go ahead and get to the question area. One of the things that seemed to pop up in a lot of the questions were questions about supplementation with some of these different types of things that you talked about. I'm going to go through, mention several. So for those of you that are watching and that did submit a question, they're going to be paraphrased so that it works for everyone. But one was about buying the NAD+, the CoQ10 and idebenone. Can you get the prescription grade? And if so, where do you buy it? And is it effective?
So most of those are supplements that you can get over the counter at a variety of different vitamin stores. I would suggest talking to your doctor about whether or not there's a particular formulation that might work for you better, and talking about whether or not there's side effects of certain ones and whether or not they could interact with your medicines before you start any sort of supplement. Because we tend to think of supplements as being benign, but there's still chemicals that we're putting in our body. And so you do want to make sure you're talking to your doctor about it. But as far as where to get them, places like GNC, Costco, a lot of places now are have a lot of these supplements readily available, and they're not overly expensive, but definitely talk with your doctor before you start anything new.
Another question was, can urolithin A supplement help with mitochondria by replenishing new mitochondria and replacing the old?
I have not heard of that one. I'm not familiar with it.
There we go. What are the mito DNA disorders that are being treated with the nucleotides?
So right now, TK2 deficiency is the one that is under investigation with clinical trials. There's also a clinical trial, I believe, happening out of Canada, looking at some of the other DNA maintenance disorders. So off the top of my head, I could list 5 or so. But again, I think most people who have a mitochondrial DNA maintenance disorder know that they have one. If you don't, you could just look up your defect and it should tell you, or you can ask your doctor if that's what it is. But they're really the disorders that affect the ability of the mitochondria to DNA to copy itself or to maintain its stability over time.
Thank you. Another question popped up, and this is an interesting way to think about it. I am often informed at our hospital that mitochondrial disease patients are not eligible for liver transplants. What are the applications for that, and what a patient need to know to go to their doctor in terms of considering such a thing?
Yeah, so the Mitochondrial Medicine Society wrote a paper a few years ago now about the safety and efficacy of organ transplantation in patients with mitochondrial disease. So having a mitochondrial disease in and of itself shouldn't disqualify anyone from any sort of organ transplantation. We frequently have people come to our hospital for second opinions because they have been told that they're not eligible for some form of transplantation. And sometimes there are times when they're not, because they're not eligible for some other reason, but solely having a mitochondrial disorder should not disqualify you from any form of transplantation.
One question that popped up that was interesting to me, and again, I'm going to paraphrase this a little bit to make it very, very clear. So people with LH1 plus often have a mitochondrial myopathy component to their disorder. Do they qualify for any of the trials that are currently happening? How does that work? Would they be disqualified because of the LH1 piece, or how does that go?
So that's a great question. So I love that question. So clinical trials are very specifically designed for a single purpose, and that is to test a therapy. And one of the things that's so important about doing a clinical trial is making sure that you can answer the question at the end of the day. And so a clinical trial is not designed to be inclusive of everybody. So they will very frequently limit who's allowed in and who's not. And so they have what's called eligibility criteria and exclusionary criteria. And so depending on what the clinical trial is, what it's designed for, what they're trying to test, each clinical trial will have its own list of eligibility criteria and exclusionary criteria. You can't always predict what you will be eligible for or what you won't be eligible for based on your diagnosis or your symptoms. You really just have to look at each individual trial to see whether or not you would be eligible for that. And almost all trials will have a coordinator or a study contact that you can reach out to. So if step one is to identify a trial that you think you might be interested in, and step two is then just to reach out to them and say, hey, this is me. Am I eligible? And usually they're going to ask you some questions and they're going to answer those questions. So you can't ever answer it in general about trials or in general about types of trials. It's always specific for a clinical trial.
And I encourage our families that if they're going through clinicaltrials.gov or have found a trial that they're interested in on the UMDF website through our clinical trial finder, if they're having trouble understanding what they're reading and they need somebody to help them go through that, please know that you can reach out to me at margaret.moore@umdf.org, and we're happy to make that happen and kind of walk you through the steps, because we know it's complicated. And we know that, exclusion criteria, inclusion criteria, all of it is a lot. So we're here. Just know that.
I actually want to say one other thing too, while we're saying that. One of the things that I think a lot of people don't know is that a lot of clinical trials, not all of them, but a lot of them have financial support for travel. So I don't ever want anyone to think I can't do a clinical trial because I can't afford to go. Call, ask, email, reach out to the UMDF. There's also grants sometimes. NORD offers grants for people to help travel to clinical trials. So you never know if you don't ask.
Right. That's a great way to kind of view it. Another question that came through is, and I think this varies depending on where you live, but is mitochondrial replacement therapy happening yet?
So the mitochondrial augmentation therapy is under clinical investigation, and it is not commercially available anywhere yet. The mitochondrial replacement therapy is a treatment that's being used for prenatal. It's a prenatal therapy for people who are undergoing, trying to get pregnant. I don't know the appropriate way to say that, who are trying to get pregnant. It is not available in the United States. It is available in some other countries.
Here's a like, I think, a broad topic. The question is, do these clinical trials require that everyone has a mitochondrial diagnosis? What about those that haven't, found their gene yet? And I think just talking through that a little bit and why it's so important that the people that are included are very specifically diagnosed. Can you kind of address that piece of things? Because I know it's hard for our families that haven't found their gene yet to not be able to participate.
Yeah, so most of them do require you to have a genetic answer. And in saying that, we know that, 30, 40, 50% of people with mitochondrial disease that we know have mitochondrial disease do not have your genetic answer. So I'm sad in a lot of situations that we cannot include people in clinical trials who don't. And I work really hard with a lot of our families to try and continue to search for that genetic answer so that we can include them in trials. The why. So in the United States in particular, when you're trying to prove that a drug works, there's a lot of reasons to do that. One is because as a patient, you don't want to be taking a drug that doesn't work. As a doctor, I don't want to be prescribing a drug that doesn't work. But probably the most important reason is the FDA won't approve a drug that we can't prove to them beyond a reasonable doubt works. And in order to do that, there are very specific and rigid requirements that they have. And one of them is making sure that you have a very narrow and well-defined patient population. That generally involves people of a certain age, people with certain symptoms, people with a certain diagnosis, and they're very strict on making sure that the patient population is very uniform, and as uniform as it can be. For a lot of clinical trials, people all have to have the same genetic change. And one of the challenges in mitochondrial disease is that it's so hard to find enough people with the same genetic change to do a trial that it would be exclusionary. You in a lot of situations, you couldn't do it. And so the way that people have gotten around that is by saying, okay, what if we just take people with a genetic change and we stick with just that population? And so that's the main reason, is because of the rigor that's required. There's also one of the things that as physicians we've recognized in the last 20 years is that a lot of people who have a clinical diagnosis of mitochondrial disease as we are identifying their genetic change, we realize that it is not in a gene that affects the electron transport chain or primary mitochondrial function. And they end up having a secondary mitochondrial functional defect. And those patients might not be appropriate to put in a trial. And so we don't want to mix the population in the clinical trial without having that solid confirmatory genetic piece. And so those are the main reasons that it is usually required to have that molecular or genetic diagnosis.
Okay, we have time, I think, for just another question or two. This is more of our vocabulary lesson here. What does expanded access mean?
Oh, I love that. So expanded access. So there are drugs that are approved by the FDA for use in the United States. So, and this is a United States thing. So that are approved for use in the United States that are, okay, let me start. So when a drug is approved for use by the FDA, they give you what's called a label, and it tells you exactly who you can prescribe that drug for. So you can prescribe it to people with high blood pressure who are between, who are over 18 years of age. And as a doctor, if I have a patient who's 16 who has high blood pressure, I am allowed to write a prescription for that patient, even though they're not 18, because that's an FDA approved drug. And I believe that that patient would benefit from it. And so I can write it what's called off label. And I'm allowed to do that. What you can't do is prescribe a drug to someone that has never been FDA approved for any indication. And these drugs are called investigational products. And they are also regulated by the FDA. So these are medicines that are under clinical trials or under clinical investigation, but they've never been approved for any indication by the FDA. They're only investigational drugs. And we are not allowed to prescribe those to patients in any way unless they are on a clinical trial or an expanded access program. So what we can do as a physician is we can ask the people who own the drug, which is the company, and the FDA, if we are allowed to offer that drug to a patient who is not eligible for the clinical trial, because we believe that the patient would benefit from the therapy. And that's called expanded access, or sometimes it's called compassionate use. So we say to the FDA, we say, look, I have this patient that I think would benefit from this therapy, and there's enough evidence that the drug is safe and that it might work, and the patient is not eligible for the clinical trial, so there's no way they could get access to it unless I put them on an expanded access program. And then we have to get the company to agree to give us the drug for free. They can't charge for it. And the FDA has to agree to allow us to use the drug in that situation. And that's an expanded access program. And it is an ungodly amount of paperwork. But it has been a true benefit for a lot of patients. So I say the paperwork piece because your GP down the street is not going to do this. This is going to be a specialist who is familiar with clinical trials and who is familiar with genetic diseases and mitochondrial diseases, because it's not something that just any physician can do, an expanded access therapy. But that's what it means. I hope that made sense. And again, it's interchangeable.
It did. It's so important. Yeah, I was going to say that compassionate use was my next question, and I knew they kind of meant the same, but it makes sense that they could occur together. I understand it now, and I think it's important for our families to learn these terms now that we have clinical trials, to understand what it means. Okay, so we're running out of time, and I want to just let everyone in the audience know that if we did not get to your question today, that we will do our best to answer it, unless the question was too specific to an individual or didn't apply to the topic at hand today. We have some upcoming events that we want to let you know about. Our next Ask the Mito Doc webcast will be on April 25th. Mark your calendars at 7:00 p.m., just like this one. And we'll be talking about endocrine complications of mitochondrial disease with Dr. McCormick and Dr. Weber. And then in May, we're going to do, on the 17th of May, a special session for the newly diagnosed. And this is going to include a host of physicians. And it's really going to be an overview. It's called newly diagnosed, but really this is the primer 101 that we want our families that are going to be attending Symposium to take to watch in advance so that they're ready for all the information that's going to be happening and all the new things they're going to learn at Mitochondrial Medicine 2023. I certainly want to thank Dr. Koenig for this really informative and helpful presentation. And Nicole, do you have any other announcements or things that you want to review?
I do not. No, thank you all so much for attending tonight, Dr. Koenig. It's a pleasure as always. Really informative, and I think our patients really got a lot out of this talk. So we appreciate you very much.
Thank you.
All right. Well, good night, everyone.